Hello there, welcome to our channel on chemistry lessons. This is an AQA A-level chemistry and we're going to look at the structure of proteins. So this is what the specification looks like here. We're not going to do the entire section in this video. This video is going to look at the structures. So the idea of the primary, secondary, tertiary structures and the bonds that are responsible for each of those structures. First up though, you know what to do. If you don't subscribe, please do. Your support is very much appreciated. Thank you very much. So what are proteins then? Proteins are naturally occurring, very, very long chain molecules. So they're huge molecules, very long chains, and you're going to find them in almost all natural substances. So humans, bone tissue, enzymes, muscle tissue, they're basically the building blocks of life. Now, don't get carried away. I'm no biology teacher, so please don't be quoting that. But they are just basically very long molecules that you're going to find in all living things, in all aspects of biological processes. That's my biology done. So if we were to zoom into a protein molecule, it wouldn't just be a very simple straight chain. If we look at the chain, what we'd actually see is something that kind of resembles this. Either they have this coil-like structure that we call the alpha helix, or what we can have is we can have what we call a beta pleated sheet, where they kind of fold backwards on each other like this. Or neither, actually, is the truth. And this would be kind of what it would look like if we zoomed in. If we zoomed in a little bit further, so we could actually see the atoms, now what we'd see is a backbone consisting of nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon. And that's what we'd see throughout the backbone. And actually what these are, it's just many, many amino acids joined together. So if I kind of break all of the amide links, if I find the amide links, here we go, that's an amide link there. So this is known as an amide group here, this carbon double bond oxygen to an NH. So if I go along this molecule and I break all of the amide linkages, there we go. We can also call these peptide links for those biology people amongst us. So what we tend to call them peptide links between amino acids, but when it's in a polymer like this, we call it an amide link. So I've actually got 1, 2, 3 different amino acids here. And as we've seen previously, the only thing that differs with amino acids is this R side chain. So what we're looking at here, this sequence of amino acids, is known as the primary structure. Zooming back out again, so we had our alpha helix, or we had the beta pleated sheets. These structures are known as the secondary structure of a protein. So the sequence of amino acids within the chain was called the primary structure. That's a sequence of amino acids. Zooming back out a little bit and seeing how the chain interacts with itself, either a beta pleated sheet or an alpha helix, that's known as the secondary structure. And zooming all the way back out, this is known as the tertiary structure, this overall tertiary structure of the full molecule. Now, we don't talk about quaternary structures in A-level chemistry, but you do talk about them in A-level biology. So I know some students will know a quaternary structure. That's when you've got several proteins interacting with each other, such as hemoglobin. Zooming all the way back in then, we're going to now focus on the bonds that are responsible for the structure. So this primary structure are held together by covalent bonds. And specifically, we call those peptide bonds between that carbonyl and the nitrogen. So it's covalent bonds here, or peptide links between amino acids. This secondary structure of your alpha helix and beta pleated sheet, remember you can have both in the same protein. These structures are hydrogen bonding. To try and explain that in a little bit more detail, I've ripped off this image from Oregon University. So big shout out to the Oregon Massive. You know who you are, my fans over there in Oregon. I'll try and make a trip over at some point next summer and sign some periodic tables. But until then, thanks very much for this diagram. So looking at the bonds that hold these kind of beta pleated sheets together, or the alpha helix together, it's the same in both cases, so that makes things a bit simple. It's a hydrogen bond between the delta positive hydrogens on the NH, so all your NHs here, they're delta plus, delta minus, that's because nitrogen is more electronegative than hydrogen, and also the lone pair on this carbonyl. So your carbonyl group again is polar. Delta plus carbon, delta minus oxygen. So there's a hydrogen bond being formed between the lone pair on the carbonyl groups and the delta positive hydrogen on the NH groups. And it's the same whether it's the alpha helix or the beta pleated sheet. It's the exact same intermolecular force. Look. And we show intermolecular forces with dotted or dashed lines. So we can see that it's an intermolecular force hydrogen bond between chains of the same protein that are interacting and giving it this either alpha helix or beta pleated sheet. So zooming back out then, we now need to look at this overall tertiary structure of the protein, of how the protein molecule interacts with itself at different sections. So this is looking at a section of a protein, and I've taken this from the website above, and what we're looking at here is how side chains can interact and how they interact with other side chains on different sections of the same long protein molecule. Now, there are different side chains, and these are the 6 that we've seen in the periodic table. Now, we've got different types of bonds that form between different side chains. So if the side chain only contains R groups, then you're going to have your van der Waals forces of attraction. And we can see that here labelled hydrophobic interaction. So these are our van der Waals. So you would see van der Waals forces in alanine, for example, between the CH3 side chains. Relatively weak. However, other molecules that have either OH, so serine has an OH group, aspartic acid has an OH group, so they're able to undergo hydrogen bonding. So we see hydrogen bonding forming or being responsible for the tertiary structure. Also, it's possible to have some ionic bonds here, look. So if you've got an NH2 side group on a lysine and the carboxylic acid group on aspartic acid, we can actually have that proton moving across and forming NH3 plus and O minus. So we have an ionic bond. Also, 2 cysteine side chains can actually form a covalent bond between 2 sulfurs. And this is known as a disulfide bridge. And that's going to occur between 2 cysteine molecules. So it's important to recognize that the tertiary structure is responsible, it's the intermolecular forces between side chains and ionic bonds and disulfide bridges. It's between side chains on the protein. And I've summarized it all in this beautiful table for you here with a description and the bonds responsible for primary, secondary and tertiary, stressing that the tertiary is side chains and secondary it's the amino acids. It's hydrogen bondings with the NH and C double bond O's on the different amino acids. And that brings this video to a close. Thanks for watching.